Thank you everyone for watching our presentation for a great opportunity to talk about our effort for the successful drug discovery using iPSC model. I'm Naomi Okugawa, scientist at Biogen. Our talk will be divided into two parts. I will give you the introduction and the transcriptomics data of iPSC-derived neurons. Then Doug Porter, scientist at Biogen, will talk about interesting data from proteomics data. Human iPSC models are playing an important role in drug discovery by providing physiologically relevant cell types, as shown on the left. Patient-derived iPSCs or healthy control iPSC can be differentiated into various cell types, then used for various assays to understand the disease of interest, to establish safety models, or to screen compounds. The plot on the right is from 2018 Neuron paper. In this paper, published small molecule screens in human iPSC-derived neurons, or NPCs, are evaluated for the assay quality and the translatability. Assay quality was evaluated by iPSC characterization, terminal cell type characterization, assay control, assay description, and assay validation. For translatability, the relevance of cell type, the assay stimulus, and the endpoint are considered. The size of bubbles or dots represents the number of compounds used in the screen. As you can see, the quality of the screen and the translatability are highly related. When the translatability is higher, the screen quality is higher as well. We believe that translatability is one of the key for the successful drug discovery, and our group is providing various human iPSC models to support various disease research units at Biogen. There are various ways to produce human iPSC-derived neurons as shown here. Commercially available neurons are easy to obtain and good for small pilot experiments. But these neurons are expensive, so not ideal for larger experiments. The second is the culture-driven method. In general, this method recapitulates developmental events and can provide good models for developmental disorders. Challenges with this method are low yield, time-consuming, and heterogeneous population. For the drug discovery purposes, we need to obtain large number of highly pure neurons. We have been using the NGN2 overexpression method established by Shudoff Lab. With this method, we can easily produce large quantity of neurons in short period of time. For patient iPSC lines, we utilize lentivirus to deliver PET-inducible NGN2 expression cassette. To obtain large number of healthy control neurons in a reproducible manner, we had generated a PET-inducible NGN2 iPSC line by CRISPR-Cas9 system. And we have been producing induced neurons, which we call it NGN2 neurons, and distributing to researchers at Biogen. The top panel shows our production timeline. We plate undifferentiated PET-inducible NGN2 iPSCs and treat with doxycycline for 3 days, then freeze down as a batch. We usually set up 24 to 50 T75 flasks and freeze down 1 to 2 billion cells per batch. Our batch QC include ICC, viability, and bulk RNAseq. The bottom panel shows our plating and culture schedule. We saw and plate neurons in the media supplemented with N2, B27, CDNF, doxycycline, and culture 1. Culture 1 is a reagent from Thermo Fisher and does work well to inhibit proliferation of glial cells in culture. As shown here, we can get highly pure neurons within a week. As I mentioned in the previous slide, we perform bulk RNA-seq for every batch of NGN2 neurons as a part of batch QC since we believe that bulk RNA-seq is relatively easy and unbiased way to evaluate neurons from each batch. To better understand our NGM2 neurons, we performed bulk RNA-seq and proteomics. As shown here, we thawed out cells from 5 different batches and plated into 6 well plates. We collected cells at DIB 7, 14, 21, 28, and 35 for RNA-seq and proteomics. For RNA-seq, we also collected samples from undifferentiated iPSCs, and 3 days after doxycycline induction, right before freezing down. Next, I will go over RNA-seq data. In this slide, principal component analysis (PCA) was performed to see how these samples cluster based on the similarity. PCA plot on the left is from RNA-seq data, and it shows reproducible clustering by time point. The PCA on the right is a sneak peek of proteomics data. Both RNA-seq PCA plot and proteomics PCA plot clearly shows that each time points were reproducibly clustered together, and the batch-to-batch variation was minimal, and much smaller than the time component. Here is the gene expression heat map of neuron markers, neural stem cell markers, and pluripotent markers. The marker gene list was slightly modified from 2018 paper listed on the bottom by adding pluripotency markers. Pluripotency markers on the right are expressed in undifferentiated cells, and the expression goes down with time. Neural stem cell markers are not expressed in undifferentiated iPSCs, but the expression was turned on in day 3 samples, then eventually goes down with time. Most of neuron markers are not expressed in undifferentiated iPSCs and day 3 samples, and then start expressing at DIB7 and tend to go up with time. On this slide, we selected the axon guidance pathway gene set from KEGG pathway database. We still need to look into each gene and its function in the dataset clusters, but interestingly, these samples were reproducibly clustered by time and suggest that these neurons gradually become mature with time. Next, we looked at the neural cell type markers. Since it is known that NGN2 overexpression can convert undifferentiated iPSCs into cortical excitatory glutaminergic neurons, as described in the original 2013 paper, we expected to see distinct excitatory neuron expression patterns differentiating from inhibitory neurons. The plot on the left shows the ranked abundance of transcript at DIB35. Top is excitatory and bottom is inhibitory. We expected to see that excitatory markers would be clustered on one side and inhibitory markers would be the other side, meaning excitatory markers expression and inhibitory marker expressions are exclusive. But both excitatory markers and inhibitory markers are evenly distributed, and it suggests that those marker expression levels are quite variable. On the right, colorful graphs are transcript level plotted with time. Some of excitatory marker genes goes up with time, but others go down. Inhibitory markers behave in a similar way. Some goes up and others go down. Contrary to our expectation, NGN2 neuron did not show distinguishable excitatory marker expression profiles. To examine the neural cell type marker expression profile in individual cells, we are currently performing single nuclear RNA-seq and analyzing those data. I hope we can share the findings with you sometime soon. Even though we could not see distinguishable excitatory neuron expression profile by RNAseq, we could confirm that our NGN2 neurons are surely excitatory glutaminergic neurons by electrophysiological assays. Top panel shows that our NGN2 neurons start spontaneous firing 14 days after plating and start showing bursting after 4 weeks in culture without glial cells. The bottom panel shows that pharmacological analysis, and the activity was blocked by AMPA receptor blocker and NMDA receptor blocker, but GABA receptor blocker did not affect. I believe we are able to show that batch-to-batch variation among each NGN2 neuron production was minimal. We could not see clear excitatory neuron marker expression profile, but we could confirm that NGN2 neuron has excitatory glutaminergic neuron properties by electrophysiology. Next, Doug will share his interesting findings from proteomics of NGN2 neurons physio. I think my part is done.
All right, so let's focus a little bit on the proteomics work that we have done. So as Naomi already introduced, we focused on 5 distinct time points during the differentiation time course of the NGN2 neurons. We analyzed all these 5 batches that we've been talking about before in duplicates and analyzed them using a TMT-based workflows using mass spectrometry. In total, our data set quantified close to 11,000 proteins. So we can generate 2 distinct type of readouts from those data. So one being a time course profile plot shown on the left, where you basically with very high precision can look at how protein levels change during the differentiation time course. What we can also do using the label-free IBAC method is to compare 2 different proteins and see how their cellular concentrations relate to each other. So in this case, for example, the green protein would be a 10-fold higher in its cellular concentration than the orange one. So how does the data look like? We can look at an example like alpha-synuclein, and I'm showing you here the 5 production batches and how the protein levels change with time. And as you will appreciate, they're in very good accordance with each other. However, we wanted to go further and actually quantitatively assess how consistent our production batches are. And to that end, we chose a technique referred to as variance component analysis, or VCA. So what you do there is for each of the thousands of proteins that we have in our data set, we get 50 different data points with the different replicates, time points, and so on. And we can now quantify how much of the variance for this particular protein is explained by time, by the production batch, or by other factors. And as you will appreciate, on the left side in this violin plot is that for the vast majority of the proteome, basically all the variation is explained by the time component and only very small amounts by batch. In fact, if we set a threshold of 10% of the variance explained by batch, we end up with only 22 proteins or 0.3% of our data set that exceed this threshold. So as shown on the right, there are some examples of proteins that are affected by the batch. However, they are not all of these 22 proteins are actually outliers of the same batches, and we could not find any particular enrichment of categories or biological processes. So all in all, we have an excellent batch-to-batch consistency between these production batches in time-dependent manner. Okay, so next we wanted to get an impression of how the proteome of these cells changes during differentiation. And so we just did a hierarchical clustering and divided our proteome into different clusters with similar expression patterns. And if we set a cutoff of 30% fold change throughout the time course, roughly half of the data set is actually not affected during these 5 weeks in culture. If we look into what changes, about 1/4 of the data set shows a fairly steady decline throughout the time course. And when you do a GO-term enrichment of the proteins that are affected and show this trend, they are highly enriched in transcription mRNA processing processes. Then another 25% roughly, or one quarter, show a steady increase. And these are really all proteins that are involved in anything that's related to neuronal function and also include ligand-gated receptors. There's a small subset that actually do not show a maximum at either day 7 or day 5 of the time course, but they're really small proportions of the proteome. So the great depth of our proteomics data set not only allows us to look at individual proteins, but also compare isoforms of the same proteins. So in this case, for example, there are the 2 isoforms of clathrate light chain B, and they show distinct time course patterns in our experiment. And interestingly, there's one isoform that is brain specific and one that is more broadly expressed in the periphery. And the peripheral form decreases actually with time. Okay. And another example is SNAP25. There is, again, 2 isoforms quantified. One increase in abundance and the other one stays relatively stable. And interestingly, the form that goes up is associated with strong expression in cortex during postnatal brain development. So all in all, we do not just see expression changes during the time course experiment of neuronal proteins, but we also see a change in splicing and isoform expression that are more associated with brain. Okay, a particularly interesting subset of the protein is the ligand-gated receptors, and there's a variety of different classes, and we basically see all of them represented in our data set, albeit at different levels. And the most abundant ones we find are actually the glutaminergic AMPA receptor, and also the inhibitory GABA-A receptor. So these are the most abundant ones. So with regards to glutaminergic AMPA receptors, these are hetero or homotetramers of the products of 4 different genes. And also what we see in concordance with the literature is that GluR2 is the most abundant monomer expressed in its isoform flip. The diversity is even higher with the inhibitory GABA-A receptor. So these are heteropentamers consisting of alpha, beta, and gamma subunits. And there's various isoforms consisting of different contributions of different gene products. And interestingly, the dominant form that we find represented in our data set is alpha 3, beta 3, gamma 2. And that's actually not surprisingly the most widespread form found in the brain, but it is actually limited to particular areas, including the reticular and hypothalamic nuclei. So this was definitely an unexpected finding. Okay, when we look at AMPA and GABA-A receptors, so similar to what we see in RNAseq, all of these subunits increase in abundance over time. And we were wondering again why, given that these neurons are mainly excitatory, we see these high expression levels of the inhibitory GABA-A receptor. But then to form functional synapses, you actually need particular proteins that are expressed on the presynaptic membrane. And if we look at those in particular, an enzyme that is required for the biogenesis of GABA, the neurotransmitter, this is barely detectable, whereas two presynaptic markers for glutaminergic synapses, VGLUT1 and 2, that facilitate the uptake of glutamate from the synaptic cleft, they're expressed at much higher levels. So again, providing evidence that interneural connections are almost exclusively formed by AMPA-type excitatory glutaminergic synapses. So last but not least, we were very interested in the health of our cells, and to that end we looked into particular categories related to cell death and stress. And when you look at these protein subsets, you will appreciate that most of them actually stay at a fairly stable level throughout our time course experiment, up to 35 days in vitro. However, a particular subclass, namely Casper's activating death receptors, showed a little bit more variance. And so there's a particular protein called death receptor 5 that showed a consistent upregulation throughout our experiment. Interestingly, if we look at transcriptomics data from tissues, it is a receptor that is basically not expressed in brain. So we are still wondering about this finding and whether the upregulation of this death receptor might render NGN2 neurons more susceptible to apoptosis, but we are currently looking into this. So in summary, I hope we were able to convince you that we have established a scalable production pipeline of iPSC-derived excitatory neurons for drug discovery purposes. We get very consistent neuronal maturation, and the batch-to-batch variation is really minimal, as demonstrated by various readouts, including proteomics, transcriptomics, and also electrophysiology. So typically, these cells start becoming electrophysiologically active between day 14 and day 21 in culture, even in absence of glial cells. And the neuronal network formation is mainly driven by AMPA receptor mediated excitatory glutaminergic transmission. We maintain high viability and also a continuous maturation even after 5 weeks in culture, where the maturation is not complete yet. So as next steps, we're currently conducting single nuclear RNA-seq experiments, mainly driven at basically done to answer the question whether we have a homogeneous cell population or whether there's more heterogeneity between cells, and also to get to the bottom of this excitatory versus inhibitory marker expression, whether this is limited to particular cells. And with that, I would like to thank a lot of our colleagues that helped to facilitate this work, especially the chemical biology and proteomics group and the translational biology group. And we had a lot of support from biostatistics and computational biology. And I would like to thank you for your attention.